Hey everybody, welcome back to another anatomy video. In this video I'm going to be going over the neuron model. Following the lesson portion of this video, just as in all my other videos, I will be including a testing portion at the very end where you can go ahead and test yourself, and the way that'll work, I will go ahead and throw the images up on the screen. I will point to a specific structure, ask you to name it. You can pause the video at home, hit play again, and then I will go ahead and throw the answer up, and so you can not only check that you have the term right, but you also know how to spell it as well, because you'll need to know how to do that for the exams. And that's pretty much it. So let's go ahead and jump right on into this. Okay, so here's the neuron model. The first thing that we're going to be talking about are just the main structures that comprise it, and then I'll kind of go into some of the more fine details after hitting the large structures. So the first thing that we see right here is the cell body, this whole structure right here. We also have dendrites, which are these projections coming into the cell body. A neuron is going to have multiple dendrites, but only one axon leading away, and we see that axon right here, and it continues on right here as well. So you can imagine taking this part of the axon and attaching it right here, and that's what we're going to have. So again, multiple dendrites coming in, one axon leading away. I also want you to be familiar with the fact that a neuron, and I guess I should say more specifically the axon of the neuron, although it can be microscopically small in diameter, its length can be incredibly long, several feet even. So just be familiar with that fact as well. I'm now going to discuss the different organelles that we find within the cell body, and then I'll discuss the specific parts of the axon itself after that. So to start, let's look at this structure right here, right in the very center of that cell body. This would be the nucleus. And then we just see a couple organelles in this model. The neuron is actually going to contain all of the organelles that a normal cell in the body would contain, with the exception of the centrioles, because neurons don't replicate and divide and reproduce, which is one of the reasons why if you have nerve damage, it's very difficult to repair. There's some surgeries and things like that, and medicine is coming along and finding ways to help remedy that problem to a degree, but it's still very, very difficult to do because of the fact that these cells don't reproduce. Lucky for you, though, because of this model, we don't have all the organelles on it, and so you really only need to know about two of them for your exam. Again, check with your professor to make sure that that hasn't changed since I made this video, but let's go ahead and look at those right now. We see these blue structures right here known as nasal bodies, and what they are essentially the rough endoplasmic reticulum of the cell body. And if you remember from the prior unit, the rough endoplasmic reticulum, again, which we refer to as nasal bodies here, are in charge of protein synthesis in the cell, and protein synthesis is used for a variety of reasons. I won't get into all of them, but hopefully you have some understanding of that by this point. But just be familiar with the fact that these are the nasal bodies, and they are the rough endoplasmic reticulum in charge of protein synthesis within the neuronal cell body. We then see these black lines right here, and these are meant to represent neurofibrils. Neurofibrils are essentially part of the cytoskeleton, which again, the cytoskeleton from the previous unit are used to help give the cell its shape and structure. So that's what we see right here, neurofibrils, and I mean these would exist throughout the whole cell, but on this model they just decided to draw them in these black lines right here. So that pretty much completes the organelles that we see within the neuronal cell body. I'm now going to move on to the axon, which again we saw right here, and then I want you to take this part and visually connect it right here, because that's really what's happening. This neuron that we're looking at, before I go any further, I want you to know that this is a neuron in the peripheral nervous system. And that's important to know because this cell has a different name depending on if we're in the central nervous system versus the peripheral nervous system. And since we're talking about the peripheral nervous system, okay, this is going to be called a Schwann cell. And what a Schwann cell is responsible for is creating the myelin sheath, which you see in yellow right here, that goes around the axon itself. Now at this point you may be asking yourself, well, what is the myelin sheath? And that's a good question. I want you to imagine or use the analogy of this axon acting like an electrical cord. that you would have in your house, because essentially it's doing the same thing. It's really carrying an electrical impulse from one place to another. Just as with an electrical cord in your house, we want to have some sort of an insulator around it. So for the electrical cord in your house, it's going to be the rubber that's wrapped around the copper inner part of that electrical cord. And essentially what that allows is for the impulse to travel much faster and even more specifically, more directly to where it needs to go. If we didn't have this myelin sheath, you could see the impulses would be able to get out and maybe travel in different areas other than right straight down the axon. And again, it also speeds up the rate of conduction through that axon. The specifics of it are more physiology, so if you're taking physiology right now, you'll probably have a better understanding. For this course, you just need to know that the impulse is helped, or sped up, I guess I should say, by these areas right here, known as nodes of Ranvier. And what nodes of Ranvier are, they're essentially these gaps in the myelin sheath, which allow the electrical impulse to travel in jumps, like this, as opposed to having to travel straight through. So you would have a node of Ranvier right here, it would jump to this node of Ranvier, jump to this node of Ranvier, and it just allows for quicker conduction. Again, that may seem a little confusing. It has to do a lot with physiology, but it certainly is the case. So, if you're interested in figuring out more about that, you can actually go online, or on YouTube, I guess I should say, and go to Khan Academy, type in Khan Academy, K-A-H-N, type in nodes of Ranvier, and he actually gives a fantastic video explaining that. Again, it has to do a lot with physiology, so just be prepared to have some physiology terms thrown at you during that video if you're really interested in breaking that down a little bit more. So again, though, we're looking at a peripheral nerve, okay? And if this was a central nerve, we would not call this cell a Schwann cell. It would actually be called an oligodendrocyte, and that's just an important thing to keep in mind. You will see questions regarding that brought up on the lecture exam for sure, so make sure that you can differentiate between the fact that a Schwann cell produces the myelin sheath in the peripheral nervous system, whereas an oligodendrocyte produces the myelin sheath in the central nervous system. If you need some way to remember that, if you can just remember SP and OC, that might help you out. SP for Schwann cell, peripheral nervous system, OC for oligodendrocyte, central nervous system. That's just a quick way off the top of my head to help you remember it. You can develop your own way as well, but it is something that you're definitely going to need to know for the exam. Inside of the axon right here, we also see these organelles, and these are mitochondria. They look very similar, if not identical, to the mitochondria that you see in the normal cell model that we, again, had in a previous lesson. And so these are the mitochondria, right? They are in charge of producing the ATP for the cell to operate. And I really don't have much more to say about the mitochondria. That's pretty much what you need to know about them. Moving back for just a second to the Schwann cell, on the very outside of the Schwann cell, in this white region that we see on the outside, is a structure known as a neurolemma. And what a neurolemma is it's essentially the husk of a Schwann cell. And let's use real quick here the analogy of a piece of corn, which I'll throw up right now for you. So there's a piece of corn, and the husk is the outer part. So imagine that inside here is where all the corn is, right? And then on the outside we have that husk. And that's essentially what the neurolemma is. So to continue on with that corn analogy, I'm going to talk here about the endoneurium, which the endoneurium is just a sheet of connective tissue that wraps around the entire axon. They've cut it away here just so you can see all the structures, but the endoneurium, that connective tissue layer, would be around this entire axon as well. And so in relation to the corn analogy that I gave you, imagine if we wanted to cook that corn, okay? So again, we had the Schwann cell, which was the corn itself, right? The myelin sheath in here, and we have the nucleus of the Schwann cell right here. We have the husk on the outside, and then we wrapped the entire piece of corn in tin foil so that we could cook it. We're obviously not trying to cook our neurons, but in terms of that analogy, I think it fits, and it might be a nice way to recall this information for the exam. Just think of this corn analogy that I gave you, and that might help. The final topic that I'm going to discuss in this video is this structure right here, known as a synaptic end bulb. And for the lab portion, that's all that you need to know, that this is a synaptic end bulb. But for lecture, I just kind of want to break this down a little bit more as to what's happening. Whereas we have multiple dendrites that carry information in towards the cell body to be processed, we have one axon that's going to leave the cell body. However, this axon is going to split up at the end into things known as axon collaterals. And essentially, if again we're using that electrical cord analogy, imagine if that electrical cord was able to fray out at the end and essentially release its electrical impulse into multiple different wires. That's kind of what we have happening with the axon collaterals. And I have an image up on the screen right now kind of showing you what an axon collateral is. And so at the end of those axon collaterals, which we see right here, they turn into what we call synaptic end bulbs, which again is the lab term that you're going to be tested on. And it's called the synaptic end bulb because it's at the end and it forms a bulb. And this is where we have the synapse between one neuron and another neuron right here. And so this would be the synaptic end bulb from a presynaptic neuron, and this would be the dendrite of the postsynaptic neuron. Again, pre versus post, because we have the synapse is that small gap, which you can't even really see on this model, in between where this axon ends and this dendrite begins. So it makes sense we would call this the presynaptic neuron. Okay, I guess you could say the axon of the presynaptic neuron, and this would be the postsynaptic neuron. Okay, specifically this is the dendrite of the postsynaptic neuron. Another thing to keep in mind here, and again this is all lecture material, and I'm pretty sure this is fair game for lecture. Again, see with your professor if they'll actually be potentially testing on this. But all that we see right here is one type of synapse. There are multiple different classifications of synapses. This right here would specifically be called an axodendritic synapse, which makes sense because we have an axon synapsing with the dendrite, okay, of another neuron. We also have, again, several different types, three main ones though. We have other synapses known as axosomatic, which we could actually see right here. Somatic refers to body, so somatic makes sense because we have the axon synapsing with the cell body, not a dendrite, okay, of the other neuron. And then we also have synapses known as axoaxonic, and they are not shown at all on this model, but it would essentially be when an axon actually synapses with another axon, which does happen as well. I'm not going to go into more detail than that in this video. You've got enough stuff on your plate as it is for the testing portion here, but just be familiar with that fact that there are multiple different types of synapses. I've laid out the main three, but there are even more than that, I think that's getting kind of ahead of the game at this point, so just be familiar with those three. And with that, I think that's pretty much it. So let's go ahead and move on to the next portion of this video where you can test yourself. Okay, so there's the lesson on the neuron. Again, now we're going to jump right into the testing portion. And I mentioned at the very beginning of the video how that works. Another thing to kind of help you out too, if you're not familiar with YouTube, you don't necessarily have to hit the pause and play button. If you just hit the spacebar button, it'll actually pause the video for you. And then you can just hit play by hitting the spacebar again. Just kind of save you a little bit of time. So best of luck, and let's go ahead and start that right now. Okay, so to get started, please name the three main structures that make up a neuron. First, this one. Then these white structures. Again, there's multiple numbers of them. And then this structure as well. Okay, so first we have the cell body. Then we have the dendrites, which again carry information in towards the cell body. And then we have one axon, which carries the electrical impulse away from the cell body. Inside of the cell body, we see a couple different organelles. Again, we would have all the different types of organelles inside here, but we can only test you on three because that's all that shows on the model. So please name this structure right here. That's going to be the nucleus. Name these structures right here as well as what they do. These are known as the Niesel bodies, and again, those are the rough endoplasmic reticulum. They are in charge of protein synthesis inside of the neuronal cell body. Name these structures down here, these black lines. Okay, these are going to be neurofibrils, and again, they would exist throughout the entire cell body, and they are just part of the cytoskeleton to help give the cell its shape. Okay, moving on to the axon. This is the nucleus of which type of cell? Again, we're looking at the peripheral nervous system here. Okay, this is going to be called a Schwann cell. If this was part of the central nervous system, though, what would that cell be called? Okay, that would be called an oligodendrocyte. Again, remember that SPOC tip that I gave you? That should help you recall that for the exam. Name this part of the Schwann cell. It helps to keep conduction moving quickly through the axon, as well as making sure that the information or the impulse isn't escaping. Okay, this is known as the myelin sheath. Please name the gaps in the myelin sheath, which we see right here, and right here. Okay, those are known as nodes of Ranvier, and again, they help the impulse go quicker down the axon because it allows the impulse to essentially jump from one node of Ranvier to the next. Please name these red organelles inside of the axon. Okay, those are going to be known as mitochondria, and again, they are in charge of ATP synthesis, which is essentially energy for the cell. Please name this outer layer of the Schwann cell. Again, when I was doing my lesson, I referred to it as the husk, right, when I use that corn analogy. Okay, this is going to be known as the neurolemma. And please name this structure out here. This is the connective tissue layer. Again, in our corn analogy, it was the tin foil that we wrapped the entire ear of corn in. Okay, this is going to be known as the endoneurium. And please name this structure right here. Okay, this is again known as the synaptic end bulb. I did fail to mention this in the lesson, so hopefully from your lecture you can just recall this. If you can't, I'm just going to test you on it now though. This region of the neuron is essentially it's a part where the electrical impulse is going to start up again, and before it moves on down the axon. What would this region right here of the neuron be called? Okay, that's going to be known as the axon hillock. Again, I didn't go over it in the lesson that I had previous to this, but just be familiar with that term as well. If you don't see it on the lab portion, you probably will see it somewhere in the lecture exam. And so this, again, is the axon hillock. It's the point between the neuronal cell body and the axon itself. It's where the impulse is going to be regenerated, essentially, and that's where the impulse starts again. Okay, well, there it is. I hope this lesson wasn't too difficult for you. If it was, you could obviously review it as many times as you want. Best of luck on your exam, and I'm sure, see you soon in another anatomy video.